go ahead.
Just to put it in context, the last time we were trying to look at how antibody responses are downregulated, and we really saw that there were two mechanisms. One was by antigen-specific suppressor cells that are actually T lymphocytes, which act to downregulate the antibody response. So that's a lack of reactivity due to the presence of something positively inhibiting a response. And we also saw at the end of the last session that if B cells interact with antigen in the absence of helper cells, that they are inactivated. And we described the spleen cell focus assay, where animals which had been immunized to carrier KLH and irradiated and a few B cells injected into the animal, some of which landed in the spleen. The spleen was cut up into 50 pieces, and then DNP carrier KLH was added to all the cultures, and the number of B cells were added was so few that there was only one or no B cells in each fragment. And Kleinman, when he did this, found out that these B cells could be activated, if there was a B cell there, by haptenated DNP KLH, so that he got a large amount of antibody produced in the culture, so much that he could even examine it by physical chemical techniques. What he found out, just to summarize so that people are on the same page, is that if he did not give DNP KLH at the time of initiation of culturing these fragments, but 2 days later, he could still get the B cells to be activated. But if he gave during those 2 days DNP of albumin, for which there are no helper cells, the B cells were inactivated. So he couldn't then not get any response when after So this was taken as evidence that B cells, if they interact with antigen in the absence of help, lose their ability to be activated and so are inactivated. So later on, some experiments were done with the new technology of making TCR transgenic mice. What someone did, and interestingly enough, he was a fairly ambitious guy who did this for his PhD and proposed it to his supervisor, who said, "You're crazy, that's much too ambitious." So he left that one supervisor and went to another who said, "Sure, why don't you do it?" And the investigator actually got a faculty position without doing a postdoc. So Chris Goodnow. So what Chris Goodnow did was he decided that it would be interesting to make mice transgenic for a B cell receptor, rearranged light and heavy chain, that could recognize an antigen, hen egg lysozyme. So in this case, the mice, transgenic mice, would express all the B cells when they developed, would express this one same receptor. And so it would suppress all endogenous rearrangements. So all the B cells of a transgenic mouse, or this kind of transgenic mouse, have surface receptors that can recognize the antigen HEL. Technique is that the cells responding to a particular antigen aren't rare. In fact, all the B cells have got the same receptor on their surface. And so what Chris Goodnow did is he exposed these B cells in various systems, either to hen egg lysozyme coupled to sheep red cells, an immunogenic carrier, and he could activate them. Or he took these B cells, or developed systems where he took the B cells, exposed them to heneglysozyme for 48 hours, and then challenged them with heneglysozyme on sheep red cells, and he found out that they had been ablated. The B cells were no longer functional. And if he took the B cells and parked them in this situation without the antigen heneglysozyme and waited for 2 days but gave then heneglysozyme on sheep red cells, he got a good response. So you could show that the presence of the antigen heneglysozyme, HEL, in the absence of HELP, ablated the B cells. So I wondered whether there are any questions on that, and whether that people sort of followed that.
Follow along. Okay.
So I mean, it's very helpful others that Brooke doesn't have to do all the saying yes. That was a so now to summarize, we've seen that B cell responses can be down-regulated in 2 ways. And now we're going to look at T cell responses. But what's very interesting is actually that the 2 same ways have been delineated for T cell responses. So luckily, we're beginning to see patterns in how immune responses are regulated. And one of the ways that one of the things that people did or tried to address is, if I have an unresponsive state, is due to the fact that I've got suppressor cells response occurring, or is it because I've got rid of the lymphocytes which could mount such a response? And they found both cases, but they were mindful that when they didn't get a response, it could have one of 2 causes: suppression or deletion. And it could, in principle, have suppression leading to deletion, but people never found that kind of situation occurring, so I'll put it aside. So one of the things that they did was they gave A strain cells into irradiated A cross B mice, where A and B strains differ in MHC. So if they gave bone marrow from A strain mice into irradiated A cross B, then they would make a radiation chimera, as we've seen before, and the A-type cells when you do this, you find out that on the whole, the A-type parental cells developed in an A cross B environment can't respond to B cell antigens, B MHC antigens. And so they wanted to know whether that or due to inhibitory cells, and they found out it was due to deletion by doing limiting dilution analysis. So in a normal mouse, we've discussed limiting dilution analysis. If you look at A-type cells which can react against B MHC, which you find out by limiting dilution analysis, and if you take A lymphocytes which have developed in an A cross B environment, you find out that the frequency is less than 1 in 100,000. So it's been depleted by at least 100-fold. And so, so this really showed that some form of unresponsiveness could be due to deletion. So are there any questions on that? So people wondered whether deletion could occur in natural tolerance. So one of the things which was very relevant to the studies is the fact that normally, as we've seen, there are two types of class 2 molecules, IA and IE. It turns out that there's a black strain of mouse which has a non-functional IE chain, the alpha or the beta, I can't remember which, and that means it doesn't express IE at all. So people immunized this strain, which is IE deficient, with IE molecules from other strains, and they found that most of the T cells that responded to IE used a particular V beta gene. So if you remember, the beta chain is made up from VDJ elements or VJ elements. I can't remember which, but it's done by recombining these elements. And it turns out that there's a particular V beta 17, which for some reason has affinity for IE, the IE molecule of other strains. And so when you immunize C57 black with V beta, or with IE, you find out that all the T cells express, or most of them, this V beta 17 T cell receptor. Do people understand what I'm saying? Thank you, brother. So what people did is they looked in thymic development, and they found out that the proportion of V beta cells, when you go from double positive to single positive, did not change in C57 black mice, the proportion of cells which were V beta 17. But if you took a mouse that had IE, then if you looked early on in T cell development at double positive CD4, CD8 T cells, you found a certain fraction had V beta 17, and that when you got to single positives, CD4 cells, single positives, they'd gone down considerably, a lot. And that was dependent, they could show, on the presence of IE in the thymus. So that really told them by a marker that the T cells developing in the thymus, which had the potential to respond to IE, were ablated due to the presence of IE in the thymus. So are there any questions on that?
Can you explain the last part again? Summarize it.
Okay. So what they did is I'll do it with a twist. What they did is they took C57 black mice, which have no IE, and they made C57 black mice which had IE because they had actually made it transgenic for the non-functional IE chain. So now it expressed IE. So you have two mice which are genetically identical, except one doesn't have IE, and one has IE. And in the mice which did not have IE, if you look at the proportion of cells which express V beta 17 as T cells develop in the thymus, it's a constant proportion. But if you look at the mice which have IE, the early cells which are generated, which have a functional receptor, they've got a higher proportion of T cell receptors bearing V beta 17 than the mature ones, which have gone through negative selection because IE is present. And so the V beta 17 cells, which are immature, which then interact with IE, are negatively selected. And so the mature thymic cells, CD4 cells, have a lower proportion of cells which express V beta 17. Did that help?
Yeah, thank you.
Okay. Two types of experiments came together, series of observations, to make people realize something about the activation of T cells, CD4 cells, and CD8, but CD4 particularly. And the first was something that we briefly talked about, but not very much. And that was when you do transplantation across MHC barriers, you find out you get rejection very fast in less than 10 days. But if you take a graft, a thyroid graft as it turns out, and you expose it to high oxygen tension for 2 days, the cells of the graft seem to be okay. And then you transplant it into an MHC incompatible recipient, and the graft isn't rejected for months. So something's happened when you expose it to high oxygen tension. And people thought that there must be a particular cell type in the graft called a passenger leukocyte, which was caused to die in high oxygen tension. And it turns out that this passenger leukocyte does exist. It's called an antigen-presenting cell, and and people really found out that the typical cell, the important cell in the graft, was an antigen-presenting cell by taking a graft, culturing it for 2 days in high oxygen tension, getting rid of the passenger leukocytes, and then giving it into a recipient who is MHC different. So supposing the donor was A, of strain A, and the recipient strain B, if you now take APC from strain A and you put it into a recipient B strain, which has got a graft which is tolerating and not rejecting, because the graft was exposed to high oxygen tension for 2 days, if you take A-type APC and put them into that B recipient, so it looks like this antigen-presenting cell is responsible for causing rejection. And it turns out that when you look at an MLC, mixed lymphocyte reaction, there are cells which have the MHC antigens. Now let me back off a little bit. Supposing we do a mixed lymphocyte reaction between strain A lymphocytes and strain B lymphocytes, and we block the strain B from multiplying by irradiating them, or let's say by irradiating them. So B will be the stimulators, and the A lymphocytes will multiply in response to the B stimulators. If you do that, you find out that you get tremendous proliferation when you take A strain spleen cells and B strain B cells, which are then irradiated. But if you take some other B strain cells, which have MHC antigens on their surface, you don't get any proliferation, or very, very little. So that meant that there was something about the spleen cells which are different from the other types of cells, B-type cells, which cause stimulation of the A responders. People, I can't draw this on the board, or I could if I'd set up a board, I suppose. But do people understand what's going on roughly there?
Mm-hmm. Yeah.
Okay. So what they found out in the long run was that the APC, not only does it have class 2 molecules and present the antigen, which can be a peptide from ovalbumin, or it can be actually allo-MHC. In other words, a class 2 molecule which is from another strain, but that the APC has got a molecule called a co-stimulatory molecule, and the T cell has a counter receptor called counter receptor to the co-stimulatory molecule, and that the activation of a T cell, a CD4 cell in particular, requires not only signal 1, the TCR interacting with the peptide MHC complex, but this co-stimulatory molecule or signal 2. So people began to realize that you needed 2 signals to activate a CD4 cell, but we'll come back to that. So did people sort of understand roughly what was going on? And then, yeah, okay, and then people decided that that APC were really complicated, because not only did they have class 2 on their surface, but they had at least one co-stimulatory signal, if not several, these B7 co-stimulatory molecules. So what they did is they made artificial bilayers, purified a class 2 molecule, put it, the class 2 molecule embedded in the lipid bilayer, exposed the lipid bilayer to an overpeptide that could bind to the groove of the restriction class 2 molecule. So then they would just have what the T cell receptor could bind, nothing else in the lipid bilayer. And they took T cell clones, cells from a T cell clone, which was specific for that peptide in the context of that restriction element, over in the context of the class 2 molecule. And they took these cells in a very rested state, so they hadn't been recently stimulated. And if they took and they gave the peptide in the APC, they got tremendous proliferation. But if they took the peptide, exposed it, the lipid bilayer with the restriction element in it, so the peptide would bind to the groove of the restriction element, and expose the T cells to just the peptide MHC ligand, they got no proliferation. So obviously that would show that to get proliferation, you needed something more than signal 1. But what was more is if they did that for 2 days, exposed the T cells for 2 days, then they found out that if they took those T cells and tried to stimulate them with a peptide on a normal APC, the T cells had gone dead on them. So this gave rise to the idea that if you generate signal 1 alone for 2 days, the CD4 cell is inactivated. Just as we've seen for B cells, if they interact with antigen without a second signal coming from a helper cell, the B cell loses its ability to be activated anymore. So this gave rise to the idea that you require 2 signals to activate CD4 cells. Are there any questions on that? So I'm sorry, I should have told you that on page 183 are these experiments I just described in figure 11. So yes, please.
In the first scenario where there was lots of T cell proliferation, you had
Yep.
the MHC complex, the antigen. What was there in the first one?
Okay. So what you do, thank you for that question. So what you do is you take a source of APC and you add the peptide that can bind to the groove of the MHC molecule. Now, what happens actually is that you've got about 100,000 class II molecules on the surface of your APC. And they all have peptides in their groove. But those peptides come off the groove. Half of them come off in about 24 hours at that rate. So even if you incubate it for a few hours, quite a few peptides come off the MHCs. And if you've got the right overpeptide, which the T cell receptor will see when that peptide is bound to a class II molecule, in the culture, that overpeptide will bind to the groove of the class II molecule, and then the T cell receptor can recognize it and get signal 1. Does that help?
Yeah.
Did you understood what I was trying to say?
Yeah. Those are on
So that's
antigen-presenting cells, right?
Yeah, with the antigen-presenting cells. And the antigen-presenting cells, as we've just seen, have got the B7 molecule on their surface constitutively. And so there's that recognized by a molecule called CD28 on the T cell. And so so the T cell will get the costimulatory molecule and divide, and you can block the interaction between the T cell and the APC by adding antibody to CD28, which blocks CD20. That you can get activation of the T cell, because now it doesn't get the co-stimulatory signal, but only signal 1. And in the notes, I won't go into it because I don't think there's time, but in the notes, there's a very neat application of that to treat people who are at end-stage leukemia. Essentially what you do is if you are on end-stage leukemia, so you've had chemotherapy, so you have no lymphocytes left, you're going to die. What they did is they took bone marrow cells from an MHC incompatible person, and there are always T cells contaminating the bone marrow. So if you gave the bone marrow with contaminating T cells to the end-stage leukemic patient, the T cells would launch, and did launch when they did it initially, a graft versus host response, which would kill the patient. I mean, it's pretty grim, this. But what they managed to do is they took the bone marrow and they gave APC from the recipient and blocked co-stimulatory interaction for 48 hours, got rid of the T cells which could react against the recipient, and then gave the bone marrow, and they had a miraculous cure for these end-stage leukemic patients. So that's described in the notes, and it's very exciting when basic studies give rise to this kind of medical intervention in such a end-stage case. So let me see, where am I going from here? So we've seen a way of eliminating T cells, CD4 cells, by blocking the second signal, co-stimulatory signal. So in a way, there's a bit of similarity here between what we know about B cells and T cells. So now I also said that people had found out that there were ways of negatively regulating T cell responses by suppressor cells. And one particularly interesting example is shown actually on figure 30, page 187. So I'm going to paint the scenario so that you can understand the logic behind these experiments. So people had been very interested actually in the 1960s in cell-mediated immunity, and they had found out that if you give a low dose of antigen, you can very often induce cell-mediated immunity in the form of delayed type hypersensitivity. So do people know what I mean by delayed type hypersensitivity?
Yep.
Do you want me to explain what I mean by delayed type hypersensitivity? Okay, I'll go ahead. What they found in the 1960s is that if they gave a low dose of antigen, they could induce delayed type hypersensitivity. If they gave in the same way a higher dose of antigen, they induced antibody. And when they induced antibody in a mouse or guinea pig or whatever, it was a very general phenomenon. If they then came along and tried with a low dose to induce delayed type hypersensitivity in an animal which was making antibody already, no longer induced cell immunity in the form of delayed type hypersensitivity. So what they said is that the immune response to that antigen had been deviated into a humoral antibody mode. So do people sort of understand what's going on here?
Yeah.
Okay, thanks. The fact that you could not induce delayed type hypersensitivity could be for various reasons. One could be that when you get an antibody response, you actually generate some cells that turn off a cell-mediated response, prevent a cell-mediated response from being developed. And in fact, that turned out to be the case. And it turned out that these were antigen-specific CD4 cells. So if we go to figure 13, on page 187, we've got a situation where a recipient, if they're not immunized, if they're immunized with an antigen B, they get DTH to antigen B. And it says DTH2 in the last but one column, and it should be to C. So that's missing in the figure. I just noticed it now. DTH to C and DTH to B. So if you give B, you get DTH to B. You give a low dose of B. You give, in line 3, you give C, a low dose of C. You get DTH to C. But if you take a low dose of B in group 2 and you give cells from an animal that's been primed to make antibody to B, if you give those cells, you don't get DTH to B. And these are antigen specific, because if you take in line 4, you take cells primed to B, they don't affect the DTH response you get to C. If you give a conjugate of B and C, so you link B and C together, you get DTH to B and C. If you give cells primed to B in group 6, and you give BC as a conjugate, you don't get DTH to B or C. But if you give cells in group 7 primed to B and you give B and C separately, not linked together, you get DTH to C but not to B. So all this shows that when you have an antibody response, you've got antigen-specific T cells, which can inhibit a DTH response. And these T cells act by linked recognition, just like you need linked recognition to get hapten-specific B cells and T cells to help, for the helper cells to help the hapten-specific B cell. That is shown by group 6 and 7. So in group 6, you give the antigens linked, and in group 7, they're unlinked. When they're linked, the B cells, the cells specific for B, can inhibit the DTH response to B and C. When they're unlinked, the cells primed to B can only inhibit the response to B, but not to C. So that's quite a mouthful for me to say, and probably quite a lot for you to digest mentally. So are there any questions on that? So
it's pretty straightforward.
Okay, good. We've seen now, it's actually getting very interesting, and actually this is a part where I really wish I could talk to you in person in a classroom, because we're going to begin to see things coming together, all these mechanisms and how they make physiological sense, and we're going to be addressing the major questions of the contemporary field. So to go, before we actually get quite that far, I just have to note somewhere, page 187. Yeah, okay, I left something out, which was on 180, around about 186. Let me just try to, probably a little bit further back. Yeah, it's a 185. I'm sorry, I missed something out on 185. But I think we very briefly covered this possibly in class. There was a situation where people looked at a CD8 response to an antigen called QA1B. And QA1B is a very weak antigen. So what they had is they had two strains of mice which were genetically identical, except that one mouse had QA1B, and that the allele coding for Q1 locus, the other mouse had Q1A. So they're taking Q1A, Q1B cells and trying to prime the Q1A mice against QA1B. QA1B, sorry, it's really confusing. Is QA1B, QA1A. We're taking QA1B and trying to get a response in QA1A mice. And the antigen is very weak, and you can only get priming against QA1B in QA1A mice if you have a carrier, and that carrier is the male antigen. So you give QA1B female cells to a QA1A mouse, and you don't get priming. But if you take QA1B male cells, you get priming against QA1B. The male antigen has to be on the same cell as the QA1B antigen, because if you have a mixture of cells, one of which has QA1B and the other one has HY, you don't get priming. And it turns out that if you give QA1B B by itself, you actually inactivate the CD8 cells, which can recognize QA1B. So that's all a bit of, again, it's best to read that quietly by yourself, but the conclusion is fairly simple. It's that in order to prime QA1B CD8 cells, you require and these helper cells turn out to be CD4 cells. Now, a cat come, so I'm going to have to remove my cat. Say hello. Hello, everyone. There's a cat. So, just as we found that you need to activate B cells, you need helper cells. It turns out that to activate or prime CD8 cells, you need helper cells, recognizing a carrier. You need link recognition. And so we are seeing a real general pattern here. Are there any questions on that? I think actually, yes?
Once they are primed with the
A1B.
Yeah. Can just a QA1B like female cell then be added and like get a response as well, or does it always have to be?
Well, it's a little bit, it's described in the notes. So what they looked at priming by seeing whether they could get cytotoxic T cells after they primed with the cells and going with QA1B in culture. So that's why it's priming rather than looking at just cytotoxic T cells. So they're trying. They had an assay for priming essentially, which was an in vitro assay. They did not get cytotoxic T cells out without the priming step, so they used this priming step as an assay for what you require to get priming. Does that make any sense? All right, so, but I mean, these are all details and obviously important, but they give the picture now that we've got CD4 cells required to activate B cells. In the absence of helper cells, B cells inactivate B cells. Sorry, in the absence of helper cells, antigen can activate B cells. The same seems true for CD8 cells. So this tells us that the fate of B cells and CD8 cells on interacting with antigen depends on helper cells. If you have helper cells around, you get activation. In the absence of helper cells, you get inactivation. So CD4 cells are really the conductors of the orchestra. So the real question is, what causes their activation or inactivation? And we're going to come back to this as a very major question. There's a lot of controversy in the field. And in order to make things clear, I'm going to give you my view. I really disagree with the major models that have been espoused for the last 30 years. And I'll try to explain why. But you have to realize that I'm giving you my perception rather than the common perception of what's going on. So I think what we'll do just one other thing here, and that is we'll go to the last thing we'll do today is we'll go to the very beginning of the next session. Oh dear, I always get messed up here. so what I'd like, okay, I'm just going to very briefly explain something which is, if you're doing research in the field, is really important. And it's important in a lot of interpretation. So it takes days to get a response. When we think about what's happening when we activate a B cell, there could be very many stages in the activation. In fact, the helper cells act on the B cell by membrane contact, and then they make cytokines, and there are a whole series of steps that occur. So when you have a lot of steps that occur, let's say you have 3 steps for the sake of argument, it actually turns out that when you look in the spleen of a mouse, there are a lot of responses to gut flora. And so there are B cells, there are antibody producing cells, a lot in the spleen, even in the spleen of a mouse, which has not been immunized by an immunologist. In an adult mouse, the responses to gut flora, so they're antibody producing cells. So if there's 3 steps that a B cell has to go through before it gives rise to antibody producing cells, in a normal spleen, you have cells which have gone through no steps, step 1, step 2, and step 3. And what's very important is if you take out B cells from a spleen, you've got some which have gone through step. So if you allow it to go through step 3, and only step 3, but not step 1 and step 2, let's say step 3, the last step just involves cytokines. You add a cytokine, and you find out, I don't need T helper cells at all, I just need this cytokine to activate the B cell. But actually, you're only then looking at the last step. And really what you want to know is what is required to go through step 1, step 2, and step 3. And a lot of confusion in the literature occurs because people don't appreciate this. They say, look, I only need a cytokine to get antibody producing cells. Therefore, there's no need for T helper cell, B cell contact. Did I make that clear? Okay. So I'm going to give you an analogy, because supposing you're taking, you want to take a donkey, use a donkey to take a lot of your luggage through a village in which the width of the path varies a lot. If you're wise, you'll go through that path and find the narrowest point in the path and not load up the donkey with a load that's wider than that path, than that width, the narrowest width. Otherwise, when you get there, you're going to have to unload the donkey, let it go through and load it up again. So if you've got a series of steps, and one step is very difficult, then going through all the steps will be governed by the most difficult step. So in fact, you often find out that there are different steps. One requires link recognition. Another one, the link recognition isn't anything like so important or strict. But to go through all the steps, you need you have to have link recognition. So the T helper cell helps a B cell in different ways, some of which require link recognition and others don't. To go through the whole series of steps, you'll need link recognition. So did the donkey analogy and did this make sense? Okay. All right. So I think we'll leave it at that in case other people have classes. And I won't summarize the on Wednesday, anything. I will give an opportunity for people who've gone through to ask me questions on what I've gone through. But I'm going to start off with figure 1 on page 192, which is the beginning of bringing things together concerning our understanding of self-tolerance. So we're really getting into integrating things together. So I'll call it a day for now. All right?
Thank you.
You're welcome. Bye.
Bye-bye.